I want to spend the next couple of slides trying to make sure that you don't get confused when you see amino acids drawn in different ways. So this is how I showed a generic amino acid molecule in one of the first slides for this chapter. I said the generic amino acid molecule always has an amine group, it always has a carboxylic acid functional group. Those two things are connected by a carbon in the middle called an alpha carbon. The alpha carbon always has a hydrogen, and then it always has something else attached in the R position. And very often students will look at this and think, well, this is how I will see every amino acid drawn for the rest of my life. However, this is just one of many different ways that you can draw an amino acid. This is another way. It's a little bit confusing because you were introduced to this way first, but you can see everything still. Here's the amine functional group down here now. It's pointing sort of southeast. Here's the carboxylic acid pointing a little bit northeast. Here's the alpha carbon. Here I've drawn the R group on the left and the hydrogen on the right. But this is just a rotated version of this molecule. And it's going to be difficult, but after a little bit of practice, you'll be able to get it. You'll be able to look at this molecule here and say, oh yeah, it's an amino acid too. It's just another way of drawing this thing. And you can draw them many different ways. I can draw the amine group on top, the carboxylic acid group on the bottom. I can draw the R group on the side. Here's the alpha carbon again, and then here's the hydrogen. But these are all just different ways, different rotated ways of drawing an amino acid. There's something else that I'd like to point out to you because it is very often shown in biology texts. This is another way of showing the amino acid. Here they're saying this is the side chain. Here I'm drawing the R group or the side chain to the north. This is the amine functional group, the alpha carbon. COOH is chemistry shorthand for a carboxylic acid group. So if you didn't know any better and you came across COOH in a textbook, you might not know what it means, but it's basically chemists and biologists have agreed that writing the letters, writing the atoms in this order, COOH, is shorthand for writing for a carboxylic acid group. The reason that this was dreamed up is it was probably dreamed up around 100 or so years ago when it was difficult to actually draw this in printed documents. In other words, to typeset this type of information was difficult, so people basically said, let's all just agree on a way of writing this on one line where we all know that we actually mean this. And they came up with this agreement, and so if you ever see COOH written down in a molecule, this is just chemistry shorthand for saying a carboxylic acid functional group. So that's the beginning of, you may see amino acids drawn in any of the ways shown on this slide. There's one additional wrinkle shown on the next slide. In most of our body fluids, and most fluids that are predominantly made of water, the carboxylic acid group and the amine group are actually charged. They are ions. And what I mean by that is here. Here is my generic amino acid, and here's the carboxylic acid group, here's the amine group. In reality, because this is an acid, it is capable of donating this hydrogen. And when you drop amino acids, for the most part, into liquids like water, this ends up getting donated. And this is actually what the amino acid looks like. This carboxylic acid has already donated its H. It's H plus actually, and because it donated an H plus, this carboxylic acid, or what used to be a carboxylic acid, has a negative electrical charge. And this amine group, this nitrogen, is actually capable of accepting one additional H plus. And so when you put amino acids into water and solutions that are mostly made of water, under most conditions the amine group accepts a third hydrogen plus ion, and because of that it becomes positively charged. So even though I have been drawing amino acids like this throughout the beginning of this lecture, where the carboxylic acid is not charged and the amine group is not charged, for the most part amino acids in our cells are electrically charged. The carboxylic acid has lost its H plus and it ends up being negatively charged. The amine group has gained an H plus and is positively charged. You don't need to know this term. There's a term for molecules that are electrically neutral but have positive and negative charges that offset each other. It's called a Zwitterion. It's a German word. Again, you don't need to know it, but it's mentioned in your book. You may have noticed in one of the previous slides, the amino acids were being drawn in this way. Here's the carboxylic acid group, but it has lost its H+, which would have been here, and this oxygen is negatively charged. Here's the amine functional group. It has gained an H+, and because of that, it's now positively charged. So for the most part, amino acids, when they are in water-based liquids like our cells, they tend to have these negative and positive charges. So if you see that, you should be aware that this is just another way of drawing an amino acid. Here comes another summary slide. Before we forget, amino acids are connected to each other, or attached to each other to make polypeptide molecules or protein molecules. So those connected amino acid molecules are sometimes called polypeptides, sometimes called protein molecules. There's a subtle difference between polypeptide and protein molecule. We are not going to get into that subtle difference, but just be aware that I'm cutting a very tiny corner, but for the most part, polypeptide means the same thing as protein. What do proteins do? So I'm sure everyone here has heard of proteins, right? You're supposed to eat a lot of protein to build muscle and that type of thing. What do proteins do at the molecular level in our bodies? They do pretty much anything and everything you can think of. They can provide structure to our cells. You can think of them as being like posts and beams of the cell. They actually give the cell three-dimensional structure in many cases. Some protein molecules, you can think of them as being workers. They are enzymes that catalyze different chemical reactions that take place in the cell that are important for our survival. Some examples of different types of enzymes: there are protein molecules that carry other molecules around in the cell, and I'll show you an example of that in a little bit. There are protein molecules that make duplicates. They copy our genes when our cells divide and make sure that each of the new cells gets one copy of our genetic material. So there are proteins that are involved in doing that. There are proteins that are involved in making our muscles move. There are proteins that carry oxygen from our lungs to other tissues in the body. This particular protein is called hemoglobin. You may have heard of it, but that is a protein molecule whose job is to pick up oxygen in the lungs and take it to other tissues. And many, many other sort of functions in living organisms. If there's something that's being done, there's probably a protein that's, if there's something being done in your body, there's probably a protein that is helping to perform that job. So one of the punchlines I want you to come away from this slide with is this: that different types of protein molecules have different jobs. So there are certain types that do one job. Hemoglobin is one kind of protein. It does this. There's a different type of protein molecule that helps muscles move, etc., etc., etc. So as far as human proteins are concerned, the estimate is that there are about 100,000 different types of protein molecules that humans can make. Now, they don't make all 100,000 at the same time in every cell, but each cell is assumed to be capable of making about 100,000 different types of proteins. Each of the 100,000 different types has a different job. And each type of protein molecule is different from each other because it has a unique sequence of amino acids. And what I mean by sequence here, sequence just means the order that the amino acids are attached to each other in. So in other words, all of these protein molecules, all 100,000, are different from each other in the fact that they have the amino acids attached to each other in a different order. So if you attach the amino acids to each other in one particular order, then you make one type of protein. If you attach certain amino acids to each other in a different order, you'll make a different type of protein. So, and it's the side chains, it's those R groups that make each type of protein molecule unique. It's the ordering, the order that the amino acids are connected to each other, and the order that the side chains come in that makes different protein molecules different from each other and makes them do different jobs. So this is basically the why of who cares. We were talking in detail about different side chains, and some being hydrophobic, and some being hydrophilic, and some being charged. Well, it's the order that you attach those side chains, and if you attach, certain ones next to each other, that ends up making each different protein molecule do a different particular type of job in and outside of the cell. So I have an example here. There's going to be a video. This particular image here is an image of a frog embryo. So this, what I'm circling here is one large frog embryo, and I'm going to show you a video in a moment. So here is the frog embryo again. It is frog embryo very early in development. So this, what I'm circling here, is one particular cell. This is a different particular cell, a third cell, so you can see individual cells. The yellow material in each cell, this yellowish material, is the DNA in the cell. So this is the chromosome of the cell. This is another chromosome. And the blue material is one particular type of protein. The blue material has been decorated with a substance that makes this particular protein essentially become fluorescent blue colored under the microscope, and so it allows you to see this protein doing whatever it does when you look at the frog cells under the microscope. And this particular protein, its job is to basically yank one copy of each chromosome to the new cell when the cells divide. So these cells are going to divide, and what you'll see is this blue protein is basically going to pull on these chromosomes, and it's going to yank some of the yellow material in this direction, and there's going to be a new cell made here, and it's going to yank some of the yellow material or the chromosomes in this direction, and there's going to be a new cell made here. And you'll see that taking place in some of the other cells as well, but I think this one is a decent one to focus on. So here we go. Here are the blue proteins, and they are going to pull the chromosomes apart, and then this is going to become a new cell, and this is going to become a new cell as well. And so the idea is that that is one particular type of protein, one of thousands of different proteins that are running around in those cells. You can't see the other ones because they haven't been decorated in a way that makes them emit a fluorescent blue color under the microscope, but they're there. You're only it's nice in the fact that you're only focusing on one type of protein, and that type of protein you can sort of see it doing its job. So just to show you once again, these blue proteins are pulling on the chromosomes, and they yank one set of chromosomes to one new cell, and they yank the other set of chromosomes to the other new cell. So that's an example of one type of protein doing one particular type of job in the cell. Again, just to emphasize, the blue stuff that you see in that video is just one type of protein molecule. There are many other types of protein molecules running around in those cells. You just can't see them because they're not decorated, and they're doing other types of jobs in those cells.